Hello, my name is Michele Francis. I'm a researcher at the University of Stellenbosch in the Soil Science Department. Today I'm going to talk about permanent carbon storage associated with organic matter in soils. I will explain how the process works and give some examples where this occurs and draw parallels with the situation in the Spekboom Thickets. Firstly, what is permanent carbon storage and why is it better than organic matter or biomass? Vegetation removes large amounts of carbon dioxide from the atmosphere as carbon dioxide is transformed through photosynthesis into biomass. The accumulation of biomass in the soil under these areas is extremely valuable in terms of storage of carbon. However, this carbon is eventually released again back to the atmosphere as carbon dioxide during the decomposition and oxidation of organic matter in soils, unless there is an intermediary step which is able to capture and store the carbon in a more permanent form. This more permanent form is the form of soil inorganic carbon, specifically the mineral calcite in dry soils. Carbon is a commodity. Here's a selection of the last 6 months. It's been the best performing commodity of 2021. The value of carbon per hectare for permanently sequestered carbon should be much higher than the price per hectare for simply accumulated organic matter, which itself is not to be undervalued. One of the ways which is able to convert organic matter into a more permanent store of organic carbon is the Oxalate carbonate pathway. This is a biogeochemical pathway that links atmospheric carbon with the accumulation of the mineral calcite in unexpected geological settings. For example, acidic red tropical soils where the pHs of around 5 makes the precipitation of calcium carbonate otherwise impossible. Carbon dioxide enters the system through photosynthesis by oxalogenic plants, where it combines with calcium to form calcium oxalate crystals in the plant. These insoluble calcium oxalate crystals remain trapped within plant tissues until they enter soils as leaf litter and are worked into the soils. In soils, oxalotrophic bacteria use calcium oxalate as an energy source. This reaction produces alkalinity, which is the key to the subsequent formation of calcium carbonate in the form of the mineral calcite, where calcium ions are available. The source of the calcium ion is very important, and much of the vegetation in these areas where the calcium oxalate pathway has been seen are naturally enriched in cations such as calcium and magnesium. It's very important that there be no calcite in the parent material of these soils, because a carbon sink is effective only if the calcium sequestering the carbon as pedogenic calcite does not originate from an inherited source. Only the first step sequesters the carbon in calcite. The subsequent dissolution of calcite and its reprecipitation, in whichever location it then finds itself, emits a carbon dioxide molecule so that the carbon is exchanged but not sequestered in the later steps. The very important thing is that the soils be non-calcareous to start with. We could see this process of carbon capture happening in a sacred forest remnant in Togo. A sacred forest is believed to be inhabited and protected by gods, which means that the local communities have very strong rules prohibiting timber and game harvesting in these forests, which protects them over many generations. And so these sacred forests are the last remnants of ecological niches which have otherwise been removed from the region. And very importantly, the vegetation in the sacred forests are extremely high in oxalate. The accumulation of organic matter and leaf litter under the sacred forest is striking in contrast to the surrounding soils. The surrounding vegetation is mainly influenced by agriculture, typically woody savanna, cleared by burning, with very low crop yields associated with low soil fertility, and the soils are badly affected by erosion. This is reflected in the extremely different organic matter, nitrogen, and phosphorus levels in the soils. Most striking is the difference between the sacred forest and the surrounding soils in terms of soil pH. The sacred forests are rich in organic matter, the soils are slightly calcareous, and they have a pH of around 8 that reflects this. There is also a lot of calcium oxalate in the plant remains, and also biogenic calcite associated with this. The following slide has a lot of information on, but I'm going to talk you through it. We made some orientated thin sections of intact soil pieces so that we could have a look at exactly where the calcite is forming. This plain polarized light image shows the sacred forest soil is very porous and comprised mainly of plant remains in various degrees of decomposition. There you see them different shades of brown, and associated with that are many mite excrements. Oributted mites eat soil organic matter and are generally a sign of soil health. There's a full spectrum of microfauna activity happening in the soils. Using crossed polarizers allows us to see the calcite. These are these spangly birefringent crystals that you can also see in the plain polarized light image, but not as clearly. They are very strongly associated with organic matter in the soils, suggesting that their formation is intimately associated with the presence of the organic matter. The surrounding soils, in comparison, have no faunal activity, no organic remains, no life in pores. The pores show a lot of clay illuviation. Some of the darker colors there are dissolved organic matter and a lot of quartz and feldspar grains. There's no calcite, there's very little organic matter. The soil is a pretty much dead soil. So how does this relate to spekboom? Spekboom has recently been found to have extremely high quantities of calcium oxalate in the leaves. The reason is it uses this as an internal carbon dioxide source under drought stress, because it means that it doesn't need to open its stomata to allow an exchange of gas, which would simultaneously cause a loss of water. So by shutting the pores in the leaves and using this as an internal carbon dioxide source, it is extremely advantageous in dry conditions. If you have a look at a cross-section through the Spekboom Thicket species, an example in the Baviaans Kloof Spekboom Thicket, you see that quite a few of them contain oxalate, the big one being the spekboom itself, where the leaves contain the calcium oxalate crystals. In the tree, where the wood contains calcium oxalate, you wouldn't expect that wood to form a large part of the leaf litter on the soil's surface. But nevertheless, it has calcium oxalate, and some of the grasses as well have variable to high oxalate contents. Sinitemba Mchunu did a master's thesis at the Soil Science Department at the University of Stellenbosch, which compared thicket which was divided into two categories, spekboom and non-spekboom matrix. The study was located at the Renosterhoek farm near Kirkwood. What we can see is a highly significant difference in pH in the soils underneath the spekboom. let me talk you through these graphs quickly. The one on the left shows depth going down into the soil against the organic carbon. The categories, blue is thicket, non-Spekboom thicket, grey is the cleared areas, which were presumed to be thicket, and yellow are the Spekboom, in the Spekboom thicket. And what you can see is, as you would expect, the organic carbon between the cleared areas and the thicket is considerably lower in the cleared areas at the surface. But as you get deeper down in the soil profile, you find that the difference decreases, which is to be expected because at 25 centimeters deep, there's not a lot of leaf litter. So most of the effect of the organic carbon is happening in the top 5 to 10 centimeters. If you have a look at the pH, same soil sample, same depth, we find that the open areas, which presumably were a Spekboom, but it's very hard to tell now because it's obviously been cleared, but the Spekboom itself, all the soils have a pH around 8. And at this pH, you can expect equilibrium with soil calcite. The non-Spekboom, the soils underneath the non-Spekboom vegetation in the thicket, has a considerably lower pH. This difference is very high in the shallow soils, which suggests that it is a leaf litter effect, and at 30 centimeters depth, nearly the differences tend to even out. This is a very strong indication that the oxalate carbonate pathway can be happening in the Spekboom soils, and that the oxalate content in the leaves is being transferred to the leaf litter in the soil, and carbon fixing is being occurred in the soils associated with this leaf litter. If we go back to the value of carbon, we calculated for the soils in the sacred forests in northern Togo that they store over 200 tons of organic carbon per hectare in 15 centimeters thick soil. If you take the price of carbon in 2030 in Canadian dollars, we have almost 40,000 Canadian dollars per hectare land value, carbon value, buried in the soils. But there also is nearly 9 tons of recalcitrant inorganic carbon that has been derived from this organic carbon that is fixed into the soils per hectare for 15 centimeters thickness. And I don't think that the value of these inorganic and organic carbon can be considered to be the same. With the inorganic carbon, the carbon dioxide is permanently sequestered into the mineral when the climate is dry and the calcite does not dissolve. We're talking about a permanent sequestration. From Spekboom thicket to calcium oxalate-rich leaf litter to calcite-rich soil, how much carbon is permanently stored under Spekboom thicket? And in the areas where the thicket has been cleared but the soil would still hold calcite, how much carbon is permanently sequestered under Spekboom thickets, and how much is it worth? Thank you for listening. I hope this talk inspires interest in the true value of carbon potential in Spekboom thickets.